Hello everyone, I'm Mridul Khurana and I'm a third year PhD student at Virginia Tech, and I majorly work in the domain of AI for science using knowledge as guidance to generative models in different scientific domains. And today I'm going to talk about how we can guide, add control to generative models for studying species evolution. All right, okay. There has been significant advancement in the domain of generative AI, where we see diffusion models have kind of revolutionized the field of image generation. And nowadays, video generation as well. We are seeing too many applications of that. Diffusion models are essentially trained to step-by-step denoise from a sample random noise and generate very high diverse and high quality images. One of the most popular form of image generation models are the text-to-image models, wherein the model takes a textual prompt as an input from the user and generates an image that kind of follows the textual description. And these models have gone pretty amazing in terms of generating high resolution images. And I tried some examples using generated examples using the Stable Diffusion 3.5, which is one of the state-of-the-art text-to-image model right now. And Stable Diffusion in general is one of the most common choices for open source text-to-image models. In the recent times, we have seen that there has been a further augmentation with introduction of various control mechanisms and adding additional modalities to these models such as pose, structure, texture, depth information, etc. And in diffusion models so that we can generate more complex and faithful images which kind of align to the user inputs. And there has been many such models which such as ControlNet, text-to-image adapters, the IPR image prompt adapters, which kind of has popularized the controllable generation of the diffusion models in these particular modalities. But what is the gap? So when it comes to generating fine-grained concepts in particular animal species, diffusion models still struggle. For example, given a species name, existing models often fail to generate morphologically correct images of that particular species that precisely reflect the identity. What I mean by morphology is basically a simple relating to the structure and form of the animal. But why am I showing this image again? Like we just saw this image in the previous slide where it's just an image. It looks like a high quality image, which is an image of a lion sleeping. But if you look closely at the paws or the feet of the lion, it seems like it has six claws, like the nails, the black nails, which you see, it seems like it has six claws instead of five, which is morphologically not correct. It should have had five claws. And this is one of the state-of-the-art models which we recently have. So here lies the opportunity in AI for science to add more control to these diffusion models and essentially generate more accurate animal images. For that, we images like are generally the currency for any generative models, and recently we have seen a significant increase in the number of large-scale image repositories for animal data sets containing various different type of organisms. And these repositories are coming from various sources like museums, university library collections like iDigBio, Glenn, and also from popular citizens science data sets such as iNaturalist, which are open source. So there lies an opportunity to leverage these large-scale imaged animal repositories for training our ML models. And we can run any ML models on these images to extract latent representations that we can solve for other downstream computer vision applications such as classifying species, which is great. But what we really want is not to just say that, okay, this is, for example, a species of a bird. But what we really want to say is this is the species of the bird because we're really interested in the because of it. For example, this is a species of a bird because it has an orange belly, black coloration around the eye, and etc. And these are what we refer to as traits of the species, or in this case, particularly visual traits. And this can be for fishes, it could be the shape of the different fins, as we can see. These traits are can be like landmark points, which are known regions, and can like and can be different things. And these kind of have been measured tediously by human experts, by scientists doing field studies. But there are also so many traits which we do not know because there was limited scale of these field studies as these are very expensive to, and by the limited knowledge of what we have been looking at, where we still don't know the entire spread of species which exist on the earth. So what we're really interested is in getting feature representation which should be meaningful and should be able to solve a biological purpose, which can potentially lead us to do scientific discovery in novel traits using these biological images. Okay, so this is where we can introduce some biological knowledge to ground the learning of machine learning features to biologically meaningful concept and goes beyond improving the predictive accuracy to getting the explainability of learned features. We want to answer the why of why this species and not just this species, essentially. So this way of introducing knowledge and external knowledge into machine learning model is kind of the growing field of knowledge guided machine learning. And in this case, we can leverage the trait-based knowledge which is available to us in forms of, say, ontologies or phylogenies, which are very popular in biology, which biologists have created over the years for different species. What really is a phylogeny? So, for example, we're given of a collection of images of bird in the cub data set and cub or the COB data set for every species, and instead of assuming a flat structure in the species classes, we know that these species are related to each other in the tree of life or the phylogeny, which kind of captures our understanding of how species have evolved over time. Know that the phylogenetic tree is constructed by looking at similarity in genetic data to identify branching points where we know at the molecular level how species have diversified. And biologists use various different ways to calculate or to get to the phylogenetic tree, but essentially it is where you can understand these different nodes, and the edges on the tree are the weights where it represents time in millions of years of how these nodes have developed over the years. And one important factor to know is basically species that share common ancestors in the tree are known to be evolutionary similar. For example, the two species of the grebes, like the western grebe and the horned grebe, are more similar than the cuckoo. However, just by looking at the molecular genomic data, we still do not know the traits, what changed at every internal level of the phylogenetic tree. And this is where we could leverage image data for potentially linking features which we observe through images and molecular changes encoded in the form of this tree. This can also help us identify traits that that make one group of species different from the others, or identify the timing of changes in traits of every species along the evolutionary timeline, which is in the phylogenetic tree. The traditional way to study evolutionary traits is macroevolution, by where experts use annotations, but these are very scarce. So the opportunity we want to exploit is to see how we can scale the process of discovering such evolutionary traits automatically deriving from images and some biological knowledge. Towards this goal, we initially introduced the framework of PhyloNN or phylogeny-guided neural networks, where the basic idea is to convert the information of every image into a sequence of features where information is structured into segments for every ancestral level in the phylogeny. By ancestral level, I just refer to as going up towards the root node from the leaf nodes. For example, we can learn the first segment of features that represent what is common across all the birds in this subtree that have the gray node as its ancestor. For the next segment, we can learn what are the differences in the descendant species of, say, the purple node versus from the orange node, and then so on and so forth, going to the blue, dark green, and green nodes. In other words, these hierarchical features capture information of how one group of species have diversified from another group of species. And we call these features of sequences as imageomes, analogous to genomes, but since these are in the image space, so we call them imageomes. And for any new biological image of any species, if we can produce its equivalent imageome, we can essentially find where it is situated in the tree of life, which can be useful for detecting novel species or for finding similarities among the species at broad scales without explicitly requiring any genetic data, just by images. We take this one step further to understand what these learned feature segments mean by plugging them into generative models so that we're able to visualize these changes in traits. In particular, our goal is now to generate biologically meaningful images that follow the correct morphology. And we also ask certain questions that can we use generative models to discover evolutionary traits across the timeline? And can we create some scientific hypotheses across the tree of life which can aid biologists to discover new species or new or novel traits? To this, we now talk about and introduce the ECCV work, which is hierarchical conditioning of diffusion models using the tree of life for studying species evolution, which we also know as diffusion. And this work was a part of a huge collaborative effort from various members at the Imageomics Institute, which is funded by NSF. And to the best of our knowledge, this work kind of is the first which introduced the modality of a new modality of tree as a guidance to diffusion models. So we introduce a simple yet effective way to condition diffusion model with this phylogenetic tree or any tree for that matter. We create a hierarchical embedding that discretizes the phylogenetic tree into 4 levels, essentially creating level groups of species at the different ancestral levels, say level 1, level 2, level 3, level 4, across the timeline. Where level 1 represents the highest ancestral level, level 2 represents more fine-grained features, and level 4 essentially represents the species-specific level information. Since these trees can have very large depth with increasing number of species, discretizing it into 4 levels makes it easier to work with as graphs, kind of with an increase in number of hops and the number of nodes kind of lose information. Where group at each level represents a subtree where we can consider from going from coarse grain to fine grain representations. We get the embeddings at each of the level for a particular species, and we concatenate these embeddings to for the 4 levels and form a sequence of tokens, which we use as the input condition to guide our diffusion model. As for the diffusion model, we're using a backbone of latent diffusion models. The real advantage of using a sequence of 4 tokens that embeds the tree rather than a single class conditional embedding which can represent a species comes during the inference stage, where we can perturb the embedding space at different levels in the phylogeny, representing different time periods in the evolutionary history, which will help us visualize these changes in generated species to identify what traits which at which node it corresponds to in the tree. Oh. We propose 2 novel image-based experiments, namely trait masking and trait swapping, which unlock the potential of embedding tree as a sequence of tokens. So what are these experiments? First is the trait masking experiment, in which we can mask out a particular level embedding by replacing it with random noise while preserving all the other components. And this helps us study what traits were acquired by species at that particular level in the phylogeny. So in this case, we can just remove the level 4 information, species-specific information, and generate species which are till a particular subtree which captures information of all the species belonging to the subtree defined by E3. This experiment is analogous to the gene knockout experiments biologists do in genomics, but we're operating on the image space rather than the gene space. Second, we introduce the experiment of trait swapping, which can be thought of as analogous to the gene editing or CRISPR type experiments, but here again we are doing it on the image space. So, say a species can be defined, like for example, a pink species can be represented by somewhere flowing from the up, beige, blue node, green node, and the pink node. And where the red species similarly can be defined as the beige, blue, yellow, and red. To understand how the green and the yellow node have diverged from the blue node and to be able to visualize what were the differences this could have happened, we can simply pick the embedding of the yellow node at that particular level and replace it with the sibling node, which is the green in this case, and see the visual differences ourselves. Before jumping into the results for trait masking swapping, we just show some qualitative results of different models and our baselines. We primarily have two diffusion-based baselines, which one is the original class conditional model. Second is we use the text-to-image stable diffusion model, which where the text embedding is essentially the scientific name of the particular species, which can be like Notaurus exilis or Gambusia affinis, where we get the frozen clip embedding as the textual representation and use that to train our diffusion model for the fish data set. And we show that our generation are as good as any state-of-the-art models. To quantify the image generation quality, we report the standard metrics such as FID, inception score, precision, and recall. And the goal really here is just to show that the phylodiffusion's generative capability is at par with the state-of-the-art models. But what we're really interested is in the new trait masking and trait swapping experiments, which the graph conditioning enables us to do. We also report classification scores of generated images using a separately trained classifier. And the aim is to verify that the generated images capture essential phylogenetic information and are able to generate morphologically correct images. And we can see that a phylodiffusion outperforms the baselines for generating the correct morphology of the particular animal images. We go a step ahead to analyze the quality of the embeddings which we obtain from different models. So on the left, what you see is the ground truth phylogenetic distance between species which we obtain from the phylogeny which we have. We then compute the cosine distances of the embeddings of different models for each of the species, and we see that the hierarchical embedding resembles quite close to the ground truth phylogeny, signaling that the alignment of embedding spaces is quite good with the phylogeny, whereas the class conditional is all over the place. We see some correlation in the scientific name embedding with the ground truth phylogeny, which is mostly attributed to the presence of the genus information in the species name itself. So, coming to the trait masking results. So, the two figures on the left show you the distribution of logit scores obtained from a separately trained classifier, where we are trying to classify images generated of the species where all the four level tokens are present, where that is basically generating in images of a specific species, which providing all the four level embeddings, and the green represents what we species which we intend to generate, which the model should do, and it is doing well, right? But now, when we mask out the fourth token, the species-specific token, the sequence, and generate images for it by providing only the first three tokens, what we see is that the images generated capture common features of all the descendant species belonging to that subtree, and the logits spread across the specific subtree or the genus. This kind of asserts the claims of generating images capturing common features or traits for all of the descendant species only, and there is no information loss or information being flowing from from species belonging outside of that particular subtree. And we can also quantify this change in distribution of logits with species within the subtree and with the species outside of the subtree, and the quantitative metrics you can find in the paper. Coming to the interesting results of trait swapping and generating novel hypotheses. Okay. For trait swapping, we can select a species which we want to study. Say in this case, Notaurus exilis, which is what we see species looks like this, which can be defined belong to a subtree defined at node A. We can now swap the embedding of node A with a species belonging to node B. Essentially, this just represents a sample image for the distribution which of the images at node B. If we simply pick the embedding of node B and replace it with node A, we generate the hypothetical species, which kind of does not exist right now. But what we're really interested is what are the difference in traits it acquired. So highlighted in green are the traits which the species originally retained from the source species. So if you see, the fin structures on top and the bottom of the fish are kind of rounded, whereas the species at B, the right one, like these fin are more pointier and like a triangular shape. But we also see some new traits that has been picked from the species at the right, which is like the head shape and the absence of the barbels or the whiskers and the face. And we also see that the tail fin is kind of bifurcated into two, which is the common trait for the species belonging to subtree B. This suggests that the hypothesis that the diversification of A from B at a certain point in the evolution corresponds to the evolution of forked tail fins and presence and absence of barbels. And we can thus say that the phylar diffusion can produce results in a language that is more explainable to scientists, which enables scientific discoveries. And we can also see this through the logic scores, where the left one is when we're trying to classify the generated images before any perturbations. And on the right, we see after doing these perturbations, we see the logic scores of the source species, the Notaurus ichthylus, slightly decrease. And there is the classifier is getting confused to classify slightly more of the Notropus genus, which we borrowed the traits from. We also show results on the CUB or the cup data set for birds, where similarly the middle image represents a hypothetical image, and we can see that it is borrowing the uh, black stripe coloration around the eye from the species at the subtree P, where the beak still remains similar to the source species. We show a number of ablations in the paper, but one ablation which I want to discuss here is the number of levels in the phylogeny. So we have really looked at 4 levels right now, but the model is robust enough to work at any different number of levels at 2, 4, 6, 8. It is just a matter of a choice of design where you of how many levels of discretization you want to do and like at what nodes of the internal phylogenetic tree you want to study. And also, it is just a matter of design choice of where in the phylogenetic tree and the timeline do you want to discretize the levels. So concluding, we saw that how adding control to generative models, we can generate morphologically accurate images of animals. And we can also generate novel hypotheses around the phylogeny, which has the potential to accelerate scientific discoveries, as essentially doing these gene knockout and gene editing experiments in biology is a very time-consuming and expensive process. Biologists can essentially get a nice hypothesis of what could the images look like. And with the introduction of knowledge guided for generative models, we can see we can have a larger impact in science and AI. And for future, we plan to expand. We are expanding to a larger number of species using iNaturalist data sets, which have more than 10,000 species and 2.6 million images, and also leveraging newer architectures like diffusion transformers to scale up the process. These are the links to the project website. You can go check out. We have a Hugging Page demo where you can play around, generate weird fishes, create hypothetical species, and we're soon going to launch demos for birds and butterflies as well too for you to be able to play around with it. Yeah, thank you.
All right, fantastic. It does look like you have 3 questions in the Q&A, so if you want to jump in and take a look at those.
Okay, open. The first question is, what if you performed a selective sequence of DNA for each current species? Then use that as the description of bird images, for instance. Might that allow you to see the effect of historical changes in DNA on the feature of animal images by feeding those novel sequences of them? Yeah, that's an interesting point if I understood the question correctly. But currently, we're not using DNA sequences to use as a condition. We are relying on the phylogeny, which is kind of maybe constructed from these DNA sequences or other genomic sequences. But in the current scope of work, we did not use DNA sequences, but that is one interesting future application where we can use models like Hyena DNA or DNA bird to be able to uh, encode the DNA information better and condition the diffusion model on. Uh, okay, I think the next question is maybe not for me. Is the embedding of levels mindful of branch length? Branch length. Yes. So essentially, this is where the discretization of 4 levels or 8 levels come into picture, where more number of levels, uh, discretization happens where the depth of the tree is more, and like for example, in the CUB data set, it is 190 species, which has a larger depth. So you can have more number of levels, to discretize on, and it really depends on where you want to discretize. So branch length does not matter in that case as of now, but the nodes is kind of more important, where which are the nodes we want to study and get the trait information at.
All right, that looks like that is it. So thank you very much. Wait, looks like there's
Okay.
one more.
Are there more diversities in images generated if you swap traits with higher up hierarchy near the top? Okay, yeah, this is an interesting question. So this is one of the limitations of the work where, and this is the limitation in biology in general, where the phylogeny is defined in the way that uh if we go higher up towards the root, right, there is a lot of loss of information. And even it is harder for biologists to validate uh, basically what are the changes which could happen. And what we really saw is like we would assume that the uh, tree is kind of a balanced tree, right? But in real world scenario, it is an imbalanced tree. So going up to the root, it essentially we saw it kind of falls into the trap of learning the like the imbalanced data set and kind of gets biased towards classes which have more representation towards that node rather than the classes which are less represented for that particular subtree.